Okay. Good afternoon, everyone. Is this on? You hear me? شغال لا؟ Hello. Hey, okay. Good afternoon, everyone. شلونكم؟ One of you emailed me about the videos. I just want to clarify that I don't have access to these videos, so I'm not the one who uploads them. But I did talk to Miss Sara, and Miss Sara explained to me that there is the e-learning team, they're working on it, and she assured me that it will be uploaded, and that you should expect the upload to take a few days, I guess maximum 1 week, but it's not instantaneous. It's a whole process. It takes a lot of work uploading the videos, the records. The lectures are recorded apparently, and so yeah, I'm just here to inform you that they are working on it. Okay? معاي شباب شابات. Can I start? All right. The other thing, مشغل المايك? The other thing that I want to mention is for the midterm exam, do you know how many lectures, how many sessions are going to be with you? 11, which means everything you took before the exam. The exception would be the 2 lectures that will be given on the same week which your midterm will start. So your midterm will start November 7th on a Thursday. So the week of November the 3rd, anything after that, this will not be included in the midterm, which means lecture 12, which will be given on November 4th, I believe, and lecture 13 or session 13, which will be given on Wednesday, November 6th, those will not be included. Anything before that, sessions 1 all the way through session 11 will be included in your midterm. مفهوم؟ وياي? All right, I just want to make sure that this information, so basically everything that I will teach you will be included, but in addition, there will be 1 lecture, the first lecture that Dr. Samah will give you, this will be as well included. So 10 lectures from me, 1 lecture, the first one from Dr. Samah. Yes? Yes? I have the sample multiple choice questions. All right. So your friend is asking whether I can give you practice, I guess, in the session, correct? Yeah. What I can do at the beginning of every session, I can kind of review the previous lecture. On Moodle, I have just added a few multiple choice questions just for you to be acquainted of how do we usually, what is type A MCQ. What other exercises are there do you have in mind? Okay. We have no problem, but for me, we have the lectures. My door is open. I can absolutely, you can come in anytime. As long as I'm there, I will always welcome you. There are very few exceptions when I know that I have a meeting coming up or someone is coming over to my office. Practice this, I can imagine you might need it for if you have a lot of math equations, things like that. Practice sessions might be needed, but in our case, I really don't know what kind of, you tell me. Maybe you can give me ideas of how I can help you. شنو؟
اللي احنا فيه نوعية جدا كبيرة.
أتفهم. أوكي.
لازم نكون نحن عارفين شلون بنحل، شلون بنتدرب، لازم في ورك شيت، لازم في أسئلة، لازم نحرك ايدنا عشان نكون عارفين.
Honestly, we have never done this worksheet. All we have, like, I can do is give you a sample multiple choice question. Um, we can study this. I can discuss it with Dr. Hawazin. It's just that the way we were used to so far, teaching and instructions, there's a lot of focus on students being supported, being provided with all the references, resources, being available for questions and queries, but we don't really have. This would have been fine. We could have done this if we had this. And I asked the head of the program whether we could have something like this, but due to your schedule, it's very tight. It was not feasible. But this could have, this can be done in a tutorial session. So for the year one MD students, we typically have once a week a tutorial. Attendance is optional. And that's open. I can ask some questions, I can put some questions, I can go over the material, I can go over hardcore topics. Would this be helpful? Because this could be a recommendation. This is something we could do. I don't know, maybe in the following the midterm if your schedule permits, but I can't do it myself, like it has to be done through the program. Yeah. All right. Yes. نعم ما اسمع رافعة كذي اوكي yes your question is your friend is asking whether it's enough to study from the slides. I think if you know the slides well, you're able to understand what's written in them, and you have attended all the lectures, you were there, you know what I've said, what I've emphasized, you should be able to do well. Like we typically, at least for this level, we typically try to cover everything that needs all the hardcore topics in the lecture. We know, we are aware of the topic, we are aware that biochemistry is a very vast discipline, and that is why we try as much as possible to cover everything in the lecture and in the slides. However, I'm sure you understand that some people might not understand me or simply don't understand the slides because many of them are summarized to some extent, and that is when you are encouraged to go back to the books. You can just select the topic that you feel like you want to expand more on, and yeah, so we have a list of recommended books. There are other biochemistry books. There's the Harper's biochemistry book, which can be found on Access Medicine. So you should all have free access to this book as well. All right. So back to your question. If you can email me requesting exactly what type of worksheet you're talking about, this is something we could discuss in the department, and we can try our best to, we're always looking to improve, and absolutely, but please, if you can send me an email, what are the expectations, what do you think would help you, and we will do our best to accommodate your request. Yes, okay? All right, we learned about DNA replication, right? And we said that it's a multi-step process. We kind of together went through these steps. We talked about the different enzymes involved, the different proteins involved. We talked about the characteristics of DNA replication. We said there are some features that distinguish DNA replication. And if you don't mind, I would like to quickly go over, I feel like we spent a lot of time having some discussion, but I still want to go over a few concepts related to last Monday's session. So which enzyme is responsible for unwinding the DNA and separating the two strands of DNA prior to DNA replication? Helicase. What else can helicase do? Identify something in the DNA, within the DNA. Origin of replication. So it can identify the origin of replication. It can see that there are certain sequences that it says, oh, you know what, we can start the replication process from this site. And then once it identifies the origin of replication, it can unwind the DNA. So that is the function of helicase. And then we said that once we unwind the DNA or separate the two strands from DNA, the DNA itself has the propensity to spontaneously re-anneal or reattach, like those two strands, they re-stick to each other, reform the hydrogen bonds between the nitrogen bases. But we say we have proteins that can keep them in that position and stabilizes the DNA in that unwound, or the unwound form. What are those proteins called? Binding proteins. All right. And then we said, which enzyme is the star of the show who actually has a capacity to read the old copy of DNA and then synthesize a complementary new strand? What is it called? DNA polymerase. And we said DNA polymerase is a little bit, fussy in that they require, before they start synthesizing a new strand of DNA, an RNA primer. And we said the enzyme that synthesizes this primer is known as primase. And once we have that primer, then the DNA polymerase can start synthesizing. And the new strand, the growth direction is from which direction to which direction? 5' to 3'. What about, how does it know which nucleotides to bring in and to bind with each other? How does it know what sequence? It must be reading something, right? It is reading the parental strand, am I right? So when it's reading the parental strand, how is it reading, do you expect? From which direction? All right, from 3 to 5' direction. And we also said DNA polymerase, not only does it synthesize the new strand, it has proofreading ability, which means if it makes, while it's replicating the DNA, if it makes a mistake, it can go back and correct it. And if this does not work, there are other systems in our body that can detect these changes, these abnormalities, these mutations, we call them. And of course, if these together fail, the DNA polymerase fail to correct the mistake, then you can have problems. Either the cell with the faulty DNA will die, or it will survive. And if it does survive, it may have this abnormal gain of function, which could turn it into a cancerous, potentially, cell. I think this is enough review for a last session. There are still other enzymes. Please do go back and read them and learn about them. But because we talked about mutations and we talked about how DNA polymerase sometimes fails or sometimes it does mistakes, and it, for one reason or another, it may fail to see that mistake or it may go uncorrected. So this is an error through DNA replication, but we have other errors that can happen in the sequence of our DNA. It is not because of DNA replication, an error in DNA replication, or because the DNA polymerase failed to identify a mistake or failed in its proofreading ability. Sometimes it could be that we're exposed to radiation, we're exposed to something toxic that can impose changes and mutations to our DNA. And once it accumulates, that could lead to, again, either the cell being sick, cell dying, or cell transforming into cancer. And sometimes we simply inherit the faulty sequence of nucleotides, not because of accumulated mutations, not because of the environment, not because of radiation, not because of DNA polymerase made a mistake, but sometimes we simply have a faulty nucleotide or gene because we got this from our parents. And this is the topic of today's discussion, and that is DNA mutations. So DNA mutations occurs as a result of changes in our DNA. And this could be the sequence, or it could be, we'll come and talk about what changes in our genetic material can happen. I just want you to know, even though I'm not going to really discuss it today, you can have problems in the chromosome as a whole, or just the nucleotide, some sequence of nucleotide. the chromosome as a whole, think of it this way. You could have one extra chromosome, can you not? Do you know a condition in which someone, instead of having 46, exactly, so Down syndrome, and Down syndrome, there's one additional chromosome. So that obviously will lead to certain abnormal manifestations. In the case of Down syndrome, there's one additional chromosome 21. There are other conditions in which there's an additional chromosome 13 or additional chromosome 18. And these babies can be born. Many of them will die in the first years of life, but with the exception of Down syndrome. But others, like Patau syndrome, Edwards syndrome, they can live for a few years. But other trisomies, what they call them, or abnormalities in the number of chromosomes, they may not be compatible with life, which means the baby dies in the mom's tummy. All right, so they die in utero, as they call it. Sometimes the chromosome number is fine, but a part of it is chipped, it's broken, it's lost. That could also lead to problems. But what I'm going to talk today about, not chromosomal abnormalities, whether abnormalities in number or abnormalities in structure. I want to focus on mutations within the nucleotides. So these are very specific. So we refer to it genetic mutations, all right? So we're going to focus on genetic mutations, not chromosomal abnormalities, not abnormalities in number of chromosomes, not abnormalities in the structure of the chromosome, but rather having a normal number of chromosomes and everything, but there's within one of the genes some kind of mutation. Do you know any of genetic disease that are prevalent in Bahrain that it has nothing to do with chromosomes, so it's not Down syndrome, it's not nothing similar, but it's something that leads to disease? What do we have? Something very common. It's inherited. Sickle cell disease. So that is an example of a genetic mutation. So let us now together go on a journey of the different types of genetic mutations that you may encounter. So number one would be what is known as point mutations or point substitutions. Sometimes we just say substitution. And as the name implies, substitution, what does it mean? It means you replace one thing with another. In this context, one nucleotide will be replaced with another nucleotide. Does that make sense? So you substituted a nucleotide in a certain position within a certain gene, maybe one gene. So there's one gene, and in this one point, a nucleotide is substituted with another. Now, the outcome of this change, the outcome will depend on different things. So based on the outcome, you could either call this substitution or this mutation a silent one, or a missense, or nonsense. Do you know the genetic code? And we'll talk about the genetic code, especially in the transcription lecture, all right? So if this is your codon, let's say there's a mutation here, a substitution. T became something else. If that something else still codes for the same amino acid, will your protein change? No. So this is a silent mutation. There is a substitution, but it did not result in a change, for example, in the protein structure. Does that make sense? All right. Missense, it's the opposite. It's not the same as silent. Silent, it goes silently. We could have these mutations and we don't know of them. It's silent. It didn't result in any change in the sequence of amino acids. But a missense, it means that substitution, that change of the nucleotide from one nucleotide to another in a given point, in a given position, and that gene resulted in a different amino acid. All right? And this, it depends on the protein, it depends on many things. The outcome, understandably, is a slightly different protein. All right? So you can think of it, one example would be sickle cell disease. Technically, you still have hemoglobin, but the hemoglobin is not exactly the same. So a person with normal hemoglobin have slightly different sequence of amino acids than patient with sickle cell disease. Sickle cell disease, they will have exactly the same, except one amino acid, instead of being glutamic acid, they will have valine amino acid. All right, so that is also a point a mutation. There's another type under that. There's something nonsense. When can it be nonsense? It means that one substitution in one nucleotide changed the codon that naturally usually codes for an amino acid, changed it into a stop codon. A stop codon does not translate to amino acid. It's supposed to be at the end, where once you finish the protein, then in the codon there will be a stop codon to terminate the translation process, which is the synthesis of the protein. So what will happen here? It means your protein will not be synthesized properly because there's a stop codon in the middle. It's supposed to be an amino acid within a protein, but there's a stop codon that's telling the ribosomes to stop, don't attach any more amino acids. So this could have more detrimental effects. All of these, they fall under substitution. Understood so far? So based on how will this change the codon and whether or not it will change the corresponding amino acid, you can understand that you could have different functional outcomes that are depicted here as those three types. Sometimes, yes. بعيد ونص بعد. I'll repeat this, don't worry, this is just my introductory slides. Don't hate me. I know that I do have the tendency to spend a long time on introductions, but I really do believe a solid start will make us together go through the slides much easier. Yes, you have a question. نعم. نفهم شنو؟ العبور الجيني. Is that crossing over? Crossing over is something that, no, crossing over is a natural phenomenon. These are abnormal things that happen. These are mutations. Crossing over is a natural phenomenon that happens where two homologous chromosomes exchange genetic material during spermatogenesis and oogenesis. That is not something that I'll be talking about in this course, because the main focus here is on biochemistry and then some biochemical principles and some principles in molecular biology, which is what I'm doing right now. All right, so we said you have this gene composed of a sequence of nucleotides, and you do know that when you have a gene, it'll ultimately either code for an RNA or a protein. For it to code for a protein, it has to first be converted to a messenger RNA. And we said that every 3 nucleotide is one codon, which represents usually an amino acid, all right? So yes, you could have substitution in nucleotides, but sometimes you simply may remove a nucleotide. You lose it. So that is called deletion. Sometimes you may not replace, you don't take one and replace it with another. You don't delete. Sometimes you add, and that is called insertion mutation. So there is insertional mutations, there are deletions, there are point substitutions, which is the first one I talked about, and there is something else called trinucleotide repeat expansion. This I'll have a dedicated slide, I'll go through it. There are splice site mutations, and finally frame shift mutations. I think those 3 will make a lot more sense when I explain them with images, so let us now, and also I want to remind you, don't forget, our DNA, we can make copies. If I want to translate, I want a protein, then first I have to go to the gene located in the DNA that codes for that protein, and then I make a copy of that gene that is in the DNA. I make a copy of it in the form of messenger RNA, and that process is known as, I'm going to give you a lecture on it. What is it called? Transcription. Once I have a copy of the DNA, but in an mRNA form, that messenger RNA, with the help of the ribosomes, will be used to form a protein. So this process, going from messenger RNA to protein, is known as translation. So there's transcription followed by translation. So you can imagine if there is a mistake in the DNA, there will be a mistake in your messenger RNA because of this mutation, and this will cause a change in your protein. All right? So this could be a change in the sequence of amino acid, a missing amino acid, or it could be an extra amino acid, or simply a protein cannot be formed. This is especially the case if there's a substitution led to a nonsense mutation. This will not even create your protein. So then you will be deficient in that protein. So let's take it one by one. Pure point mutations or point substitutions, that is a change of single nucleotide in a gene, and this will result in a change in the corresponding messenger RNA. And there are two types of substitution. If you substitute a purine with another purine, or a pyrimidine with another pyrimidine, we refer to this specific type of point mutation or substitution, we refer to it as transition substitution. If the purine was replaced by a pyrimidine, or vice versa, meaning a pyrimidine was replaced by a purine, that type of point mutation, that type of substitution mutation is referred to as transversion. So transition, purine is replaced or substituted with a purine, or pyrimidine is substituted with a pyrimidine. Whereas transversion, the purine is replaced by pyrimidine or vice versa. So let's say this is your DNA. This is a codon, ATG, and on the mRNA, it'll be U, A, and C. So this would give, let's say, a tyrosine amino acid. But then if you could substitute G with another amino acid, and you will have a different messenger RNA codon. If the codon also codes for the same amino acid, this will be called what type of? Silent. If it's a different amino acid, missense. And if it's a stop codon, so it's not even an amino acid, nonsense. All right? And of course, if A is replaced by G or C with T, so the purine is replaced with another purine or pyrimidine with another pyrimidine, we say it's transition type. And just to remind you, this is as well something that I do want to introduce. This is the genetic code table. We have like 64 different codes, and each codon is composed of 3 nucleotides. And as you can see, in blue, it is the amino acid. And next to it is the codon for that amino acid. And you notice there are a lot of features. If you look at arginine, which is an amino acid, how many codons does it have? 1, 2, 3, 4. So in many cases, this we will take it in later lectures, the features of the genetic code, the characteristics of the genetic code. Because there is more than, usually many amino acids have more than one codon, so you can have substitutions that will be silent, all right? You just basically, because if a substitution resulted in another codon, but that other codon still codes for the same amino acid, you can understand that this will not really create any problems, all right? So there are 3 types of, let's say, physiological outcomes. It could be silent mutation, missense, nonsense. And I like that if, under normal condition, let's say a codon is TTC, if you look here, for that, TTC will be at the mRNA level AAG. If you look at A, and then another A, and then a G, AAG. So it codes for what? Lysine. This is under normal condition. Let's say there was a point mutation and you changed it, especially in the third position. TTT became TTT. So instead of TTC, that cytosine became thiamine. At the messenger RNA level, it's AAA, which also evidently codes for lysine. So what do you call this type of substitution? Silent. But something else could happen. You could have a mutation, a substitution here, where instead of T, you have A, which codes for UAG, which is a stop codon. So this is nonsense. Or you could have missense, and there are two types, conservative and non-conservative, but in both cases, you will have different amino acids. All right. So silent mutations are also known as synonymous substitutions, and that happens when the change resulted in the same amino acid. Did we change the amino acid? No. So I'm going to skip that because I think I explained it a lot. For the missense, yes. All right. It's the same actually that I mentioned. So basically, all right. You want me to repeat the silent mutation or all of them? Yeah, so I'm starting with silent. I'm repeating it again. And trust me, if I go to something else, like if I start talking about insertion and deletion and you still don't understand substitution, please stop me and I'll go over it again. Silent mutation, it's a type of substitution. You substitute one nucleotide within a codon with another. When you do this, you could either still code for the same amino acid, and this will be known as silent mutation, or you could code for a different amino acid, and that is called missense mutation, or you could code for nothing. That one change, that one substitution, instead of giving you an amino acid, it actually gave you a stop codon. Stop codon does not translate to protein. All right? Yes. Oh, that is a very hard question. Why is there mutation? There's so many. I don't have a lecture on this, but I can give you very briefly. Your friend is asking why it happened in the first place. So sometimes mutations are inherited. The best example would be if I like sickle cell disease. There's one substitution. It's a point mutation in the sixth position of the beta chain of hemoglobin. That substitution, instead of having at the sixth position of beta chain, instead of having a glutamic acid, amino acid, or glutamate, you have valine. And that single substitution creates a problem or creates sickle cell disease, and this is inherited. You could have mutations because of errors in DNA replication. It could be because of environmental factors. It could be diet. It could be things that are, some of them, things we control, like smoking, eating unhealthy food, exposure to radiation. And there are things that we have less control over. Maybe errors in DNA replication could be one of them. Does that make sense? So these errors occur. It could be UV radiation. It could be cosmic radiation. Air travel, air travel, there are risks associated with frequent air travel. You do get exposed to these cosmic radiation, which potentially could cause mutations. And it's not always that a mutation will create a specific disease, because sometimes it's an accumulation of mutations that could lead to disease. And there are so many diseases. Not just cancer, some of them are inherited. And in this case, it is something that like sickle cell, as I said, it is purely inherited. It's not something that, because I'm not eating healthy, then I got this substitution, right? So really, it's a whole topic on its own. All right. خلصنا silent missense. ده كنسميه synonymous. Here we can call it non-synonymous substitution. And that is because of the redundancy of the genetic code, meaning that oftentimes every amino acid, they have more than one codon, all right? So this gives the chance that potentially when you substitute, if the substitution changed the codon, but the codon still happens to code for the same amino acid, then this type of substitution is referred to as missense mutation or non-synonymous substitution. Sorry, excuse me. So if it's the same amino acid, it's called silent synonymous substitution. If it is so happen that that change gave rise to another amino acid, this is known as non-synonymous substitution or missense mutation. Of course, the result, it depends. Sometimes you could have a different protein, but it's still somewhat functional. Sometimes it is functional, but not very, it's suboptimal, and sometimes it's completely not acceptable. There are examples given here. I'm not going to go through them, but it is just a concept for you to know. Sometimes removing one amino acid with another can completely disrupt the function of a protein. We don't really see that with sickle cell disease. With sickle cell disease, there are certainly problems, but the patient can survive and can live a normal, near normal life. All right, so then the result of that the change in the protein, that altered protein, the result might be acceptable, suboptimal, or non-acceptable at all. And the nonsense mutation, we said if this substitution resulted in a codon, that is, what do we call, what type of codon? A stop codon. And that is basically a nonsense mutation. It'll lead to premature termination of protein synthesis. So what does nonsense mutation cause? It means there was a substitution and the new mutated codon happens to be a stop codon. That means that maybe instead of your amino acid having, let's say, I'm just hypothetically speaking, let's say it's 20 amino acids, but somehow this substitution created in the middle of those 20 amino acids, in the middle of it, there's a stop codon, so then your protein will not be synthesized. Because as you add amino acid 1, 2, 3, the ribosome will see a stop codon and will stop protein synthesis. So you'll end up maybe with a truncated protein or a 5 amino acid protein. And more often than not, this will not be a functional protein because it affects the function of the functionality of the protein, the structure of the protein, and so on. All right. There are examples given here, but obviously nothing to memorize. You don't need to memorize these examples. You just need, if I ask you, you need to know if it's a substitution or a deletion. If it's a substitution, is it missense, nonsense, or silent? All right? And the differences between these. This is just an example. This is giving you example lysine. Okay, this has all the examples. Like, let's say TTC, this is normal. Let's say, which codes for lysine. Let's say there was a substitution. Instead of having TTC with the codon AAG, you have a ATC DNA where the mRNA is UAG. So this is a stop codon. This codes for lysine, which is what happens in a normal condition, but this is a stop codon. So then your protein will not be synthesized, and there will be a premature termination of protein synthesis. All right. For the next few minutes, I'll talk very briefly and superficially about those other types of mutations. I said that sometimes you could have deletions, sometimes you can have insertion, and you need to know what that means. Insertion, you insert something. Deletion, you remove something. The outcome, I'll briefly talk about it later. But also there's something, this is a separate category of mutations known as trinucleotide repeat expansion. Sometimes within the same coding area, In some instances, there are a few repeats. So this could be CAG repeat. This is the most commonly one that is applicable in real life and in clinical practice. There are certain proteins, this one protein that has these CAG repeats or CAG repeats, and it's usually in a healthy population, those repeats within the coding region, within the gene, should be something small. You could have 1 repeat, 2 repeat, maybe something below 6. So in green, you are fine. You are in the normal category. But sometimes this expands, especially it can occur in families. They may have those repeats, but they're more than normal. And depending on how much of those repeats, they may or may not have a problem or a disease or a clinical manifestation. The best example for this is something called Huntington disease, but I did not mention it here. It is not something for you. It is a type of neurological disorder that is inherited, usually in the autosomal dominant method. You learned about Mendelian inheritance in school, right? That's excellent. All right. So all you need to know, trinucleotide repeats may be present in some proteins, but if they are expanded, if you have more, and there's usually a cutoff point. This cutoff point could be 40, it could be 200. That will ultimately lead to a protein that is so large and very unstable. If it's large and unstable, it may aggregate. Aggregate, it means clump together. If it clumps together, and imagine this now really large protein that has a lot of CAG, which codes for an amino acid, which is CAG, I believe it's glutamate. I need to look that. You don't need to know that, by the way. All right, CAG, it's glutamine, yeah. So if you have a lot of this amino acid, instead of having it short, then you'll end up having this really large, unstable protein. And this is really mostly seen in Huntington disease. You don't need to know the disease for this purpose. But then this large protein can clump together. And if this clumping happens, you can imagine in a sensitive tissue like the brain cells, that could lead to premature neuronal death. And that is why we say people with Huntington disease have a degenerative, neurological degenerative disease. And that has a lot of manifestation, including some kind of tremor, where the hands shake this way. So that is a very specific type of mutation that we refer to it as trinucleotide repeat expansion. So we may have these, tri means 3. So they repeat themselves. Usually it's CAG. You may have 1, 2, 3, less than 6, but some families, they may have expansion. And when you have expansion, that can cause disease. And that is why, even though this is inherited disease, but a feature of Huntington disease is that you can have variations within the family. They could be affected with different levels. Your hand raise is so distracting. So, anyways, yes, go ahead. Yes, absolutely. Yes, Huntington disease is also a form. It falls under the neurological degenerative diseases, but the mechanism, the pathophysiology is different. So there's Pick's disease, which is more dementia. There is Parkinson's disease, which is degeneration, but specifically in the motor nerves. All right, for Parkinson, it's specifically motor nerve that is affected. Here as well, it's more motor. That is why they have this rasha, this tremor. But the type of tremor is different than Parkinson's disease. All right, so expansion, not good. It's okay to have some repeats, some trinucleotide repeats within a gene, within a protein, but if you have way too much, if for whatever reason, and that can be seen in some disease, there's an expansion in those repeats, you could have a faulty problem. I'm going to talk about transcription, I think next week, next Wednesday. Is it next Wednesday? Yeah, next Wednesday. DNA has a lot of genes, right? Genes, we make a copy of them. If I need to express a gene, I make a copy in the form of messenger RNA, and that process is known as transcription, صح? And then that messenger RNA will be translated into protein, correct? That messenger RNA, when I immediately make a copy of the DNA, do you think it immediately leaves the nucleus and it's ready for translation, or does it undergo modifications, some changes? I think you took them probably in high school, and I'll talk about these in detail in next week's session. But essentially, you need to do some trimming. You need to do basically, first of all, you need to add something called a capping in one side. You need to add a tail on another side of that messenger RNA. That will enable it to leave the nucleus. But one important thing that we do is we cut something within the DNA. So, if this is your messenger RNA, immediately, please ركزوا الحين بخلص. If this is your messenger RNA, immediately after transcription, it's still not ready to be translated. You need to remove the introns. The introns, we don't want them. They're there for other purposes, for stability, so it's part of the structure of a gene. When we talk about the structure of the gene, not all the gene will code for an amino acid. Parts of it, it's considered that only the exons basically will code for amino acids. Introns do not. So when you make a copy, you'll make a copy of the whole gene, including the introns, which should not code for any protein. It's not part of the structure of the protein. So that is why we need to process the messenger RNA first. And we do it by a process known as splicing. Splicing means you cut here, remove the introns, all of them, and splice together, join together the exons. So this is a correctly spliced messenger RNA. It completely has only exons that will code for protein. فاهمين عدل? So what happens? Sometimes I could have a mutation in this area, for example, AG, GT, where special proteins cut and remove the intron. If there is a mutation, maybe a substitution, but the substitution happened in a splice site, then these proteins don't know how to cut, and they will cut the wrong way. So now you'll have the sequence of your messenger RNA is incorrectly spliced, so maybe you might miss a whole exon, a whole bunch of amino acids, a whole part of your protein, and that can cause a problem. This particular type of mutation, because it occurred in the splice sites that we usually cut, it will become unidentifiable to those group of proteins. We call them SNRPs. I'll talk about them next week. So they will no longer recognize this as an intron, and they will make mistakes in the processing of RNA. So can that lead to a mutation? Can that lead to a problem? Yes, and this specific problem, we refer to it as a splice site mutation, because now the messenger RNA, fresh, still in the nucleus, freshly synthesized, still not ready to be translated, still not ready to meet the ribosome to make a protein. We said we need to make changes. There are actually three. We add a G cap, a poly A tail. Don't worry, we'll discuss these processes. But part of the change is we remove the introns. If the splice sites have a mutation, it means I can't remove the introns accurately and properly. And this type of mutation is referred to as splice site mutation. Last but certainly not least, we still have a minute. Now I said we talked a lot about substitution, but sometimes we insert a nucleotide, or sometimes we delete a nucleotide. Imagine if you add one or remove one nucleotide, or if you add two or remove two. what will happen to your entire code? Will there be shifting? This is called a frame shift mutation. For example, so you understand what I'm saying, right? Because then everything else, if you remove a single nucleotide, all the codons will change from the point after the mutation, right? There will be a shift. So everything will become jargon, all right? Everything will become different. Whereas in substitution, the change is still within one codon. So you will only affect one amino acid. Either it's going to be the same, or it's going to be substituted with another amino acid, or it's going to be a stop codon. But you won't affect the other codons. Does that make sense? But if you add insertional mutation, you add a nucleotide, or remove, and if it's only one or two, then this will cause frame shift mutation. If it's always three, then you will add one amino acid, or remove one amino acid, because the codon is 3 nucleotides, صح? So that makes sense. So frame shift mutations, does it happen when I add 3 nucleotides? No. It has to be 1 or 2, but not multiples of 3. All right? And this is basically, if this is your normal amino, your messenger RNA, that has 1 codon, 2, 3, those are the codons, and this is the amino acid for the protein, and at the end of it, there's a stop codon. Imagine you removed the C. So now, instead of CAG being a codon, everything will shift. Now it'll be AGA, and that codes for another protein. So everything after the point of mutation will become shifted. The whole frame will shift, and everything else will change, as seen here in pink. In pink, that is the abnormal amino acids. They're not the sequence of amino acids that should be there. So this is known as a frame shift mutations. But if it's 3 nucleotides together that are inserted or deleted, you'll simply have an additional amino acid or one less amino acid. Does that make sense? Yes. All right, so this is the example here. If this is normal and you accidentally added in codon to 1T, so this will become TCC, which will code for something else. But you can imagine everything else after the second codon will be different. Thank you so much for your time. سامحونا على القصور إن شاء الله ما كانت وايد ثقيلة المحاضرة. All right. Thank you so much. الساعة 2 ودقيقة. Sorry, I'm late. طب راح الهاي. There are a few things that I wanted to say. First of all, one of you emailed me regarding the uploading the videos of the lectures. And so I spoke with Miss Sara, and because you need to know that I don't have access to these videos. I know, I didn't even know we were recorded, but well, anyways. These recordings are basically, the people who have access to it are the e-learning team, and they are working on it. So you should expect that the upload may take a few days, up to 1 week, all right? So it's because they're processing a lot of videos, and sometimes uploading such videos could take time. So please be patient, but rest assured that your request was forwarded to Ms. Sara, and Ms. Sara assured me that the e-learning team is aware of that they need to upload these videos, but it does take some time. I'm only making this announcement because I believe there was a student who asked me to upload them, so I wanted to clarify that I'm not the one who has access to these videos. The other thing, your midterm is fast approaching. Do you know how many lectures are with you in the midterm? 11. So 11 sessions. So all of my lectures from session 1 to session 10, including the first session of Dr. Samah. All right, so total 11 sessions. This means that if your midterm starts on Thursday, November 7, then session 12 and session 13, which will be on Monday and Wednesday, November 4 and November 6, respectively, those will not be in the midterm. Will not be in the midterm, only in the final. بس حبيت أوضح. واضح صح؟ All right. All right, so I'll basically start by very quickly going over DNA replication, what we took in Monday's session. And from that, we will start today's topic, which is on DNA mutations. Who can tell me how do you start the process of DNA replication? What is the first thing you want to do? So basically you break the hydrogen bonds and separate the two strands of the DNA. And which enzyme does that? Helicase. What else does helicase? What can it do? What else does it do during DNA replication? It can identify sites to start the DNA replication, or what they call the origin of replication. So they identify origins of replication. Once they identify them, they separate the two strands of the DNA. Once you separate the two strands of the DNA, you need to prevent it from reattaching or re-annealing and reforming these hydrogen bonds that were formed. And which proteins do this? A single-stranded binding protein. So they stabilize the DNA in the unwound form, all right, in the open form, all right, so you can have single strand of the DNA and you can start the replication process. Which is the enzyme that synthesizes the new strand? What is it called? DNA polymerase. And we said DNA polymerase, when it synthesizes a new strand, in which direction is this new strand growing? 5' to 3'. But then it has to read something. It has to read the parental DNA. So when it reads it, in which position or direction is it reading? 3 to 5. All right. And we also said that even though DNA polymerase is so important, it's such a star, but it's also very fussy. And it has a requirement before it can start synthesizing and replicating DNA. What is that requirement? It needs RNA, a small stretch of RNA. We call this small stretch of RNA, what do we call it? Primer. And that primer, who synthesizes this primer? Primase. And if you want to remove it later, who removes it? No. Yeah, we'll link them together. Ligase links them together. But RNase removes at the end of the process. All right, so we said that our DNA polymerase, it's in addition to being able to synthesize new DNA, it also has proofreading ability. What does that mean? Means if it makes a mistake, can the DNA polymerase correct it?
Yes.
It can. But sometimes things happen and DNA polymerase makes a mistake and doesn't correct it. And that could lead to mutations. But there are other ways in which we can get mutations. What do you think are some other ways in which we develop mutations? Yes?
Mutations can be found also in women when they get blue eyes.
In women, when they get,
The humans, when they get blue eyes. When they get the blue eyes.
yes. Oh, it's a form of mutation. So, in other words, mutations can be inherited. Am I right? Mutations can be inherited. Can you give me an example of a mutation that is very common in Bahrain that causes a disease and it is inherited from both parents? Both parents have to be carriers to sickle cell disease. That's an example of a mutation that is inherited. But there are mutations and there are changes in the DNA sequence that could occur because of things we do have control over. Like sickle cell disease, I don't really have the control over it because it's your parents, right? If they carry the faulty gene, they will transmit it to you. But there are things that I can control. Maybe bad habits. They can cause mutations over time. It may or may not, but it's linked to mutations. Maybe smoking, unhealthy lifestyle, nitrates in food, so the diet, the type of diet you consume. So these are things that you could also minimize in your life, but there are things that you absolutely have no control over. This could be exposure to sunlight, exposure to UV radiation, exposure to cosmic radiation. So there's so many things, there's so many ways in which we can get mutations, and there are different types of mutations. But today, I'll focus on genetic mutations. And there are basically, sometimes you could have a problem in your genetic material, and that problem is in the form of problems related to the chromosomes. Let's say there is a problem in the number of chromosomes. Do you know a disease where there is a problem in the number of chromosomes? There's an extra chromosome. Down syndrome. So Down syndrome is also known as trisomy 21 because there's 3 copies of chromosome 21. So this is an example of an abnormality in a number of chromosomes. Sometimes the number of chromosomes are fine, but the structure is abnormal. So we refer to this as structural abnormalities of chromosomes. I am not going to talk about abnormalities in chromosomes in today's lecture. I will focus on genetic mutations. So your gene, the gene itself that has a sequence of nucleotides, because if I have a whole extra copy of chromosome, that is a whole other level of abnormality. If your chromosome is missing an arm or missing a short arm or a long arm, that is a structural problem. It means you will lose a lot of genes that are all located in that arm. So that's different. Here, I'm really looking at the gene level, single genes, for example, and what abnormalities you can have. So these are the genetic mutations that I'll talk about. This is a summary of today's lecture. But before I go through these, I want to remind you, we have a DNA, and we said DNA is coded to codes. There are coding areas that code for either ribonucleic acids or protein. For us to form the protein, one must first make a copy of the gene within the DNA, and that's usually in the form of messenger RNA. That process, what do we call it? Transcription. Once I have the messenger RNA, all right, which is kind of a copy of your DNA, the gene within the DNA, then this messenger RNA will go to the cytoplasm, will meet the ribosomes, and This will help in the process of synthesizing protein. What do we call this step? Translation. Okay? So going from DNA to protein, first you need to synthesize mRNA. That process is known as transcription. Then from messenger RNA to proteins, that is known as translation. So if you have a mistake in your DNA, in a gene, then this mistake will occur in the messenger RNA, and this mistake will happen in your protein. Yes. قصدك الليتات؟ Sure. والله الاي سي اذا علمتوني شلون اصكه وايد بكون شاكره لكم. لان لو تعرفون ايش كثر انا افلج في المكتب. All right. So the mutations that happen in the DNA in the sequence of your nucleotides, please guys in the back, you are louder than you think, or you are louder than you expect. I can actually hear the commotion, so please keep it down. So imagine, you do know that, you know what a codon is? Yes. You do know that, right? So if you have a DNA, the DNA has sequence of nucleotides. Every 3 is a codon. Every 3 is a codon. Once again, if there's a mistake here, the codon will be different. The codon on your messenger RNA will be different. The amino acid in this protein will be different. It makes sense, right? Just to give you an idea of how mutations could have an impact. So these are basically what I'll be talking about today. The first thing is something known as point mutations or point substitution. What is point substitution? Substitution, in English, what it means, I replace one thing with another. So if one person has a substitution, this means you'll replace one nucleotide with another nucleotide. All right? So far, so good? What is substitution? So this nucleotide will change, it will become something else. That's substitution. Sometimes you can remove one nucleotide. That's deletion. Sometimes in the middle somewhere you may add a nucleotide. That is insertional mutation. فاهمين so far? وياي متابعين? So you can substitute nucleotides, you can delete nucleotides, you can insert nucleotides. All these things can occur, and they are different types of mutations. And there are other types as well, something called trinucleotide repeat expansion, splice sites mutation, and frameshift mutations. This, I'll talk about them later. These are my last few slides. So I want to now talk about point mutation, which is the first type of mutation that I'll talk about today. So in point substitution, like I said, one will be substituted with another nucleotide. The effect of doing so, the outcome of doing so, can result in either one of those 3 listed under that the substitution part. So it could either be a silent substitution, or a missense substitution or mutation, or a nonsense mutation. The next question, you may ask me, what is silent mutation in substitution? It means if I change this one nucleotide with another nucleotide, so I'll have a new codon, messenger RNA. But if the codon still codes for the same amino acid, then there will be no effect on the protein. And we refer to this type of substitution as a silent mutation. Understood? If I substitute one nucleotide with another nucleotide, so I will change the codon in the DNA, which means I'll change the codon on the messenger RNA, which means you might think one outcome is it might change the amino acid, but sometimes the codon, even though it changed, but the new codon still codes for the same amino acid. So this will not be affected. This is a silent mutation. Why can we have this? Because, ركزوا معي, every amino acid or most amino acids, they have more than one codon. If every amino acid has only one codon, it means we can't have silent mutations. But because we are lucky, not as lucky as people without mutations, but because it's in our advantage the fact that every amino acid has more than one codon, which means sometimes you could have substitution, but if this substitution led to another codon which still codes for the same amino acid, then it's silent. You won't be affected. This is silent mutation. But sometimes when you substitute one codon with another, you'll end up with a new codon that does not code for the same amino acid. So you end up with a new, different amino acid. This is known as missense mutation. تعريف عليكم. The example of this type of mutation is sickle cell disease. There is only one substitution. This substitution will lead to, instead of having one amino acid in the globulin chain of hemoglobin, the beta globin chain, instead of it being glutamine or glutamic acid, it'll be valine. So you substituted one amino acid with another. So this is a missense. The third possible outcome is when you substitute one nucleotide with another nucleotide, there's a new codon. That codon does not code for the same amino acid, because otherwise I would say this is a silent mutation. That codon does not code for another amino acid, because then I would call it a missense mutation. But if that codon actually codes for stop codon, which means it will not give you any amino acid, and it will lead to premature termination of protein synthesis, this is referred to as nonsense mutation. So point substitution has three different outcomes, and by definition, it is the change of a single nucleotide in a gene. And based on that change, there will be a change in the messenger RNA. So this will be a substitution. If you imagine that this is your DNA, and if you have ATG, the mRNA will be basically a copy of the DNA, except that it won't, a complementary copy. So it's going to be UAC. UAC is a codon for tyrosine. Let's say there was a position here, this was substituted with A. So you have a new codon, right? بس بالصدفة الكودون دي بعد it codes for tyrosine. So luckily, is this what type of substitution or mutation? It is silent. But if it codes for glutamine, what would I call this? Missense. If it codes for nothing and it's stop codon, it is a nonsense. So this is one classification for substitution based on the outcome. But there's another classification based on the change itself. If you change a purine with another purine or a pyrimidine with another pyrimidine, this is known as transition type of substitution. If the purine becomes a pyrimidine or the pyrimidine becomes a purine, this is known as transversion. If you change within the same group, transition. If you switch between them, transversion. So far, so good? All right, so these are the silent, missense, and nonsense. And just to remind you, this is the table of the genetic code. No, of course, you do not need to memorize this. But what I need you to know is each codon codes for either a stop codon or an amino acid. And many amino acids do they have one codon or multiple? Multiple. So if you look at threonine or alanine, alanine has 4 different codons. So if you have this or this or this or this, they will all code for the same. So if a substitution changed the codon, but it still remained within those 4 types, then you would still have the same protein, no problem. All right? And this is again an example of what was meant by silent, nonsense, and missense. You do not need to know conservative versus non-conservative. All right, so if you have TTC in the DNA under normal level, and let's say there's a mutation TTT, there will be a change in the messenger RNA. Instead of the codon being AAG, if you go here, A, and then another A, and then G, AAG, there's one. So it's still lysine, because AAG and AAA, still lysine. So AAA, this is the mutated one. So this is a silent mutation because it's still lysine, correct? But let's say the first one was mutated, and now you have a UAG. What is the UAG? Oh, look at that. UAG is a stop codon, so this is a nonsense mutation. Sometimes you could have another mutation that will code for another that is non-lysine, maybe arginine, threonine, something else. This is missense mutation. So silent mutation, it means that the new codon still codes for the same amino acid, right? And you don't change the protein structure. This is also known as synonymous substitution. Synonymous substitution. If it's non-synonymous, this usually gives us missense mutation. Non-synonymous meaning you will have a different amino acid. You will have a different codon. That different codon codes for a different amino acid. So it's non-synonymous substitution. So for example, if this is the normal amino acid, methionine, lysine, phenylalanine, glycine, and then stop codon, and you end the protein, that is the end of the protein. This is a normal, let's say, DNA. Let's say there is over here a non-synonymous substitution. Instead of glycine, you'll have a serine. So it's a different amino acid. So that substitution gave rise to another amino acid, and that is known as missense mutation. The best example would be sickle cell disease or sickle cell hemoglobin. So the people with sickle cell disease, they don't have hemoglobin A, they have hemoglobin S, which is exactly the same as hemoglobin A, except that in one position in the beta chain of hemoglobin, instead of glutamic acid, they have valine amino acid. So there's one amino acid that is different. And the result could be either acceptable or partially acceptable or not acceptable. That what we're trying to say is that some substitutions don't really, even though it's a difference, the function of the protein is not greatly impacted. But sometimes it is only partially acceptable, like sickle cell disease, because these patients they can't survive, obviously. The hemoglobin is still functional, but it's suboptimal, and they have complications, and they have problems, right? They have painful crises. They have other complications that could happen. And there are other mutations or substitutions that will completely affect the function of the protein and hence are considered non-acceptable. Lastly, don't forget, if that substitution led the codon to become a stop codon, this will lead to a premature termination of protein synthesis. All right? So which mutation causes premature termination of protein synthesis? Nonsense mutation. Like instead of having this as a whole protein, now after methionine, you immediately have, instead of having glycine, phenylalanine, you'll just stop synthesizing the protein. So that creates a lot of problems. Obviously, you won't be able to synthesize the protein, or you may, depending on the site of mutation, you may only be able to synthesize parts of it. And more often than not, this is less likely basically to be functional protein. My focus is really on the substitution. It's very important for me to know the difference between the different types. So transition substitution versus transversion. شنو transition? Yeah, so the nucleotides are within the same category. You replace a purine with a purine or pyrimidine with a pyrimidine. If a purine is replaced with a pyrimidine and vice versa, what do I call this? Transversion. This is one classification. The other classification is based on the outcome. I could either have no change at all in the sequence of amino acid. That is known as silent. Or I could have a different, that substitution changed one amino acid. That is known as missense. Or I can have a premature termination. nonsense. خلصنا. Yes.
الريزلت إذا كانت acceptable نقدر نعتبرها إن هي silent.
No. This is still a different protein. So it's not a silent one. Sickle cell disease. Silent means nothing is wrong with you. You have exactly the same protein. We could have, the patient could have it and could have nothing. Sickle cell disease, they have a problem. They need follow-up. They live a normal life, but they still need follow-up, and they do have complications every now and then. Yes.
الأمينو أسيد اللي بيطلع لما يكون silent mutation هو نفسه بيطلع لما يكون multi mutation.
Yes, exactly the same. Why is that? Because if you look at the genetic code, many of them, like proline, has 4 different codons. So if a mutation changed this codon to this or this codon to this, regardless of which codon it is, the end result is still the same amino acid. So this is silent. It does not create any problems. It is a mutation, but it's a silent one. It means it changed in sequence. And regardless of what caused, could be an error during DNA replication, but luckily it led to the same protein. All right. Any change in the DNA, all right, we refer to it as mutation. خلصنا substitution. Now I want to talk about a unique feature. Sometimes a gene that codes for a protein, in the middle of the gene, it has something called trinucleotide repeats. Tri means 3, nucleotides are nucleotides. What are these 3 nucleotides? It's almost always CAG, because they repeat themselves. And they code for glutamate, all right? Or glutamine. All right, so this CAG repeats, you may have it for certain proteins. Let's say it's the Huntington protein. You could have it up to a certain level. It is okay to have a repeat in that amino acid shown in red. This is your protein. And this is the CAG repeat or trinucleotide repeat. So you have the same amino acid repeated, and you will be completely healthy. But sometimes in some individuals, for some reason, they have trinucleotide repeats expansion. For reasons, maybe they inherited from their family, for other reasons that we don't fully understand, this will expand. So instead of having maybe 3, 6 repeats, they may have hundreds of repeats. So what happens to the protein? That repeated amino acid becomes so big in the middle of the whole protein, so it becomes a large protein. It impacts the function of the protein, and sometimes it makes it aggregate. Aggregate means it clumps and it precipitates within the cell. The best example of this type of mutation, this is commonly seen in a neurological disease known as Huntington disease. You do not need to know the example. You do not need to know the features of Huntington disease. It's just an example of a disease in which there is an abnormal protein so large because there's trinucleotide repeat expansion. All right, and this abnormal protein will, especially in the brain, will accumulate, will precipitate, will cause the nerve cells to die, and these patients will have different neurological manifestations, including motor problems, problems with coordination, but problems with their hands. They could have this tremor. And this is not Parkinson disease, it's a different disease, Huntington. But they're kind of within this, they fall under the same umbrella. So this is, again, if you consider that this is the gene for the Huntington gene, and it has a normal level of CAG or trinucleotide repeats. Maybe in green is the normal. You could have people with some kind of expansion in yellow, but they will be unaffected. But as that expands, the patient, there will be a cutoff value in which the patient will start having abnormal protein and will start having symptoms. So this typically runs in families. The mode of inheritance is autosomal dominant. You do not need to know these details. Did you take Mendelian inheritance in school? So you know what autosomal dominant is. So that this is autosomal dominant, but it's also unique because of this expansion feature. You could have all the brothers and sisters, they might be affected, but they could have different levels of the disease because they may have different levels of expansion. If they have a lot of expansion, they will manifest. If less, they may not show a lot of symptoms, and so on. All right. So we now know what is trinucleotide repeat expansion. So we could have trinucleotide repeats, but very little. If it expands too much, it can cause problems. There's another type of mutation known as splice site mutation. I have a question for you. When we make a copy of DNA into messenger RNA, we said that this process is known as what? Transcription. And the messenger RNA to protein, what do we call it? Translation. We will have 2 separate lectures on these 2 processes. Now, when we make a copy, a messenger RNA copy of a gene within, yes, question? I can't control the AC. There's no way, I really can't control the AC. Everything is centralized. I can't control the AC in the lecture hall. I can't control it in my office. I can't control it anywhere. The whole university, they have their own controls. I honestly don't know. But if you are uncomfortable, please do maybe talk to Ms. Sarah. She can talk with the maintenance and they can sort this out. Back to my point. When we make a copy of a gene within a DNA in the nucleus, we have a messenger RNA, right? Is it ready to leave the nucleus, this messenger RNA, immediately and go to the cytosol and start translation with the ribosomes, or does it undergo changes? It undergoes changes. There's something called 5' G capping. There is poly-A tail and the poly-A tail and the 3' side. That is some of the changes that we will discuss. Don't worry. محاضرة ثانية. But part of the changes, we need to do something called splicing. Have you ever heard of this? Let me tell you very briefly. Imagine this is your messenger RNA, which is a copy of the gene, but in an RNA form. This will have exons and introns. Those are all nucleotides. Some are exons, some are introns. The introns, we don't want them. They don't code for amino acids. Only the exons do. So part of the processing of the messenger RNA, we have to remove the introns. And this is the function of a group of proteins known as SNRPs. I'll talk about them next week, so don't worry. You don't need to know them at this stage. But the SNRPs identify certain sequences. يشوفون مثلا AG أو GT ويقصون. They cut. And then they splice. Splice means join. They will put the exons together. All right? These special proteins. Now, these special proteins, once they remove the introns, this will be a correctly spliced mRNA. It'll look this way, where only the exons are present, and this is ready to bind to ribosome and start translation, because we only translate exons. Imagine if you have a mutation in this site, the nucleotide that should be there for the SNRPs to cut and splice. Let's say there was a mutation. What will happen is that the SNRPs will not know that this is an intron, so it might keep it. Or you might lose, it might keep it, so this will affect the translation. You will have abnormal protein, or it might, you could end up losing this whole, instead of cutting here, you might cut here, and you only join 1 and 3. It'll look something like this. This is called exon skipping. This is retention of intron, whatever it is, whether this if there is a mutation in the splice site, means the splicing will not occur properly. You'll either end up with intron that is not supposed to be there, that is supposed to be removed, or you may lose introns with a bunch of exons together, all because there was a mutation in the splice site. So this is known as splice site mutation. All right? So when mutations occur in one of those nucleotides common at the splice sites, which are usually used to guide these things that I called SNRPs, if there's a mistake, the SNRPs will become confused and the splicing will not occur properly. So this is known as splice site mutation. So far, so good? Okay. The last one, I promise, I talked a lot. You know, when I also talked about, I said substitution, I said trinucleotide repeat expansion, I talked about splice site mutation, but remember I said there's deletion, there's insertion. I sometimes can remove nucleotides or add nucleotides. And if I do this, if I add 1 or 2, or if I remove 1 or 2, this causes something called frame shift, because everything beyond that point where I added or removed, anything that comes after it will become kind of gibberish. Everything else will change. Think of it as the domino effect. If you remove, let's say, I'll show you another picture. If this is your normal DNA sequence, this is codon 1, codon 2, codon 3. This codon will code to an amino acid, another, and a third. Hello? This is normal. Let's say I inserted one nucleotide here, or 2. What happens to codon 1? Did it change?
Yes.
But what happened to the rest? Will they change? Everything is shifted beyond that. So now everything beyond this site of mutation becomes something else. So you're shifting the whole frame. The whole codons will shift by 1 nucleotide. Does that make sense? فهمتوا علي? But what if you added in the middle, instead of just 1 T, you added 3? What will happen? You'll add 1 extra amino acid, but the rest will be the same, shown in blue. So you'll have, for example, glutamine, and then this added mutation because you added 3 nucleotides. So this will add 1 additional insertional, this is a mutation, and one amino acid. But the rest will become the same, proline and threonine. All right? So frame shift mutation occurs if you add 1 or 2 nucleotides, or remove 1 or 2 nucleotides. Do you agree? Okay, so this is known as frame shift mutations. So if you have 3 nucleotides as triplets, this will then result in either an extra amino acid or a missing amino acid, unless, of course, what you're adding codes for a stop codon. All right, so this is all. So to recap, these are all the mutations, the genetic mutations that I want you to know about. Just please know the differences between each, the definitions, transition versus transversion, silent versus missense versus nonsense. All these are with you. I'll stay here. I'll be happy to take any questions you may have, as we still have few minutes remaining. Yes. هلا والله.